Hey everybody, welcome back to my YouTube channel. It has been a while since I have been on here. That's definitely because my research took a bit of a gap. It was a lot of me graduating earlier than expected and I had to rush it all. And this semester's been really difficult, so I didn't have too much time to really divulge in the YouTube channel or the YouTube world, but my plan has changed. I am now applying to PA school. Starting this fall, I take my GRE. I plan on filming that whole journey of me studying, exactly what tools I'll be using to study, and taking people on that journey with me. I also will be applying to a few different PA schools, and I'll walk you through that entire application process, as well as the day in the life of a patient care technician or the day in the life of a pharmacy technician, which are the two jobs I currently am working. Now, it's going to be a lot of different things on this channel. I hope you're very interested in seeing what we have to do. But today, very, very exciting, I had my honors thesis presentation. Now, I already did this. This was this morning. I'm coming back and filming this intro a little bit later, but I hope you watch this whole thing. It's a 10-minute long presentation about what I have been working on for 2 years. It's the biobehavioral effects of neural inflammation on embryonic zebrafish through inflammation and oxidative stress. You'll see the presentation. There's a few bits where it's blurry. I do apologize about that. I actually had my mom filming it, but I hope you stay tuned and see the full 10-minute long presentation, as well as a few questions that were asked at the end. And it's not going to be too long of a video today, but if you have any thoughts, just please comment and share. And my link tree, which is linked below, actually has a link to see my presentation, as well as a link to my thesis itself. So if you're interested in seeing my thesis, or maybe making some comments on what I could do better, I mean, you could. I'm never writing a thesis again. I don't plan on it. PA school will hopefully not have me write a thesis, but I thank you guys for being on the journey with me thus far, seeing what I did back in the day to really get this far. And I hope you kind of see how what I did in those previous videos has translated into what I'm doing now. I thank you and I appreciate you, and let's get started. And we are investigating the biobehavioral effects of embryonic exposure to neural inflammation and oxidative stress. So the purpose of this study was to look at the individual as well as the combined neurodevelopmental effects of elevated inflammation combined with oxidative stress on zebrafish embryos and larvae. We used lipopolysaccharides, LPS, and acetaminophen, APAP, to induce the inflammation and oxidative stress respectively. We then measured the brain development and the behavior of the larvae using several different methods. So, APAP and autism spectrum disorder, what are they? Well, first we need to start off by knowing the embryonic stage of development is extremely sensitive for all organisms. Brain development in human embryos begins at just 6 weeks. And a recent article touted by Dr. Brower raised concerns that the pre- and perinatal exposure to APAP may result in the elevated risk of developmental disorders. When we're looking at autism spectrum disorder, we know that it results in the display of these atypical social, emotional, and perceptual behaviors in humans, as we can see from the DSM-5, which is a diagnostic manual of psychological disorders. And most previous models of APAP exposure really don't take into account inflammation as a factor, and we really wanted to focus on that factor in our study. So, what are we looking at? Well, what are the neurological, behavioral, and physiological abnormalities that may occur in larval zebrafish after inflammation is induced followed by oxidative stress. So LPS causing inflammation, followed by APAP to reduce that inflammation. Then we want to look at how does that combined effect of inflammation and oxidative stress really compare to the individual inflammation or individual oxidative stress on the larva alone. Our hypotheses were, does LPS treatment result in significantly less activity compared to the controls? We also investigated APAP treatments that would result in significantly elevated activity as compared to the controls. And then we also hypothesized that LPS alone will result in phosphorylation or the deactivation of the GSK3 beta pathway, which again is our pathway of interest for this study. Why would we use zebrafish? I love zebrafish. They have a 24-hour breeding cycle where we can begin breeding one day at, say, 7 p.m., and then the next day, we are already dosing their embryos and watching them develop in real time. Zebrafish are extremely fucated with 100 embryos per clutch when we use 2 females and 1 to 2 males. So we're able to get a very large hatch rate, as you see in this photograph over here. They are also transparent for up to 3 weeks post-fertilization, which personally I think is an amazing feature of these guys. In the top left corner are about 4-hour post-fertilization embryos, where you can see their development and cell growth. And then right here in the bottom left is about 96 hours post-fertilization. You can see all the internal organs, which is great for studying the physiological abnormalities that may occur. We also looked at the embryos for our study because APAP and inflammation induce behavior and physiological effects, as we've seen in other articles. Zebrafish also will express GSK3 beta, which again is that signaling pathway of interest we wanted to study. Now, to begin, we started off with a brand new clutch of eggs for each test that we ran. They take about 12 hours to breed, and it's very sensitive, where right at their circadian rhythm cycle at the break of night, we had to put the fish in a breeding container, and then that first break of light in the morning, we would remove the slide that's blocking the fish, let them breed, and within a few hours, we had eggs that we could test. And the embryos were collected, then incubated. With that 4 hours post-fertilization, you saw how tiny those eggs were previously, we were able to induce inflammation using LPS. We then followed that with a oxidative stress using APAP at 24 hours post-fertilization. And here we use that 96 well plate in the bottom left seen there, and this was just some parameters that we used for our dosing measures. At 96 hours post-fertilization, we ran a light-dark test. This is a very, very interesting psychological test that we were able to use on these fish to track and monitor their activity. It was a 60-minute behavioral test with 10-minute intervals of light, followed by a 10-minute interval of darkness with a UV light underneath, so the fish during that night cycle were not disturbed. However, we were still able to monitor their behavior using the EthoVision tracking camera. And the larval activity increases at that transition to dark cycle and ceases at the light cycle. So here we see a light cycle and then a dark cycle where moving to that darkness, they should be more active, and we'll record a lot more of that activity for our analysis. A camera was mounted above the plate, as seen in this bottom picture right here, and it would track each individual well, and then the automated tracking software monitored the individual larval activity. 96 embryos that are extremely small, and then one camera was able to monitor all of that. Very fascinating. We then ran what's called a Western blot analysis. It's a protein analysis to look at that desired GSK3 beta pathway, and the phosphorylation concentration was able to be seen based on size in our electrophoresis gel. The membrane is probed with specific antibodies, and we are able to bind to our target protein membrane, where we can then use our fluorescent imaging, seen in the bottom left picture, to really look at how concentrated it is. So the bigger the line and the darker the line, the more concentrated that protein is going to be. And Then we get into our analysis. What did we find? It's really cool, let me tell you. So the behavioral analysis in the EthoVision tracking software that we use, we were able to use all of these little red dots are all of the individual fish. So when we look at the comparison between our APAP and our control, we noticed that their activity was extremely similar. So leaving a fish by itself and then just giving a fish APAP, their activity is gonna not vary as much. However, we had a p-value of less than 0.001. That means there's a lot of significance. Whenever we compare the activity of LPS or LPS and APAP to our control variable, there was seen to be changes in that light-dark cycle of decreased activity. So notice how before we mentioned they should be more active during the night cycle, less active during the day. These fish just weren't active. Then we have our Western blot gels and how they looked. As you can see, we were comparing 3 different things with the phosphorylation concentration of GSK3 beta, then our total GSK3, and then our gap deep H, which is just a marker that we used. And we had 3 different trials and 3 different gels that we ran. Now, looking at the total GSK3 beta to our gap DH, we ran an ANOVA and a post hoc Bonferroni test. This revealed that there was significantly less total GSK3 beta in our APAP group, seen in that 3rd column, as compared to our LPS group in that 2nd column. So we had a p-value of less than 0.05, still indicating that significance, and it was a very interesting comparison to see with just the total concentration. However, what I found most fascinating is that when we looked at the ratio of our phosphorylated GSK3 beta to our total GSK3 beta, and running that same ANOVA and post hoc Bonferroni test, it revealed that there was a significantly higher ratio of the phosphorylated GSK3 beta to our total. And you can see that here with our LPS and APAP being significantly high compared to that control. And that's bad. We don't want to see that. So the key takeaway from this study is that LPS and combined treatments showed to be the most impactful to our embryonic behavior in the larval study. APAP did not protect from inflammation as we hypothesized previously, and the LPS may have been too high or APAP dosing too low. However, future tests will need to be ran in order to find that discovery and really play around with those dosing factors. The elevated phosphorylated GSK3 beta to GSK3 beta total ratio has been implicated in atypical neural development, such as overproliferation of neural growth and atypical glial cell function. Then the overproliferation of neural growth and atypical glial cell functions are key physiological indicators of autism spectrum disorder as seen in the DSM-5 manual, as well as other studies that we pulled to run this test. This is just my works cited. It was a very interesting way to pull from these. Y'all got any questions? Also, thank you to Dr. Mans and Dr. Joshua Harrington for all their help. I didn't acknowledge them, but they did a lot, guys. No questions. Cool. Dr. Mans.
I have a question. Yeah, what was your favorite part of this research project?
It's definitely not the breeding. I would say the brain dissections. I had a lot of fun with that, and really just seeing how they were able to develop in different ways in the abnormal structures of some of the fish, where we could see if we really hit them over the head with too high of a dose. And then just sitting in that lab, and it was it sounds weird to say, but it was calming to just sit there and do some dissections.
What would you think would be the next step in this research?
So there's actually a lot of things I discussed with the professor moving forward in this study about looking at the different dosaging, so lowering the amount of LPS that we use and maybe increasing the amount of APAP, and see if that has any indication of whether the APAP will be protective later on in the study. And then we also want to change some of those behavioral tests that we do Maybe include a few more psychological based tests.
Spend all the time you have for questions. Thank you so much.
Thank you to everybody who watched that and just showed it some love. If you have any questions, please feel free to put them in the comments below, and I look forward to seeing you guys next time. Thanks.